Dear students, welcome to Tungala online classes. So in the last video, as we saw what are nucleic acids, where they are present, what are the functions of nucleic acids. There are two types of nucleic acids. So one is DNA, another one is RNA. And DNA, its structure, the one who made the first model of DNA, right? So how is the structure of DNA? So types of DNAs, all these are the things that we have gone through in the last video. In this video, let us start with another type of nucleic acid, that is RNA, ribonucleic acid, right? So this is another type of nucleic acid. So let us go through this RNA. In the last video, at the end part, I had explained what is RNA, how it differs from DNA itself. And one more thing is, so RNA is made up of single strand, right? So single strand sequence of a single base sets of you are going to see in RNA, right? And only difference between DNA as well as RNA itself is, so there the thymine is replaced by uracil. This is also one more concept so to be remembered by you people, right? And so next one, types of RNAs. Already occurrence as well as types I have mentioned there. So one is genetical RNA, second one is non-genetical RNA. So are the two major types of RNAs that we go through, right? So the first one, genetical RNA. What is genetical RNA? So it is the type of RNA which acts as the genetical material in some of the organisms called as genetical RNA. It is a type of RNA which acts as genetical material in organisms. In organisms is called as genetical RNA. Say for example, in HIV, RNA itself is the genetical material, right? It is the genetical material. The best example for this one is HIV, human immunodeficiency virus itself, right? And the next one, non-genetical RNA means what? This is the type of RNA which does not act as the genetical material of any organism, and such kind of RNA itself is called as non-genetical RNA. Simply, it is also a type of RNA which does not act as genetical material of any organism, right? And such kind of RNA is called as non-genetical RNA, and non-genetical RNA is classified into three types. Number one, mRNA. Number two, trRNA. Number three, rRNA. So these are the three major types of RNAs that we come across in non-genetical RNAs. First one is mRNA. What is this mRNA? mRNA itself is called as messenger RNA. Itself is called as messenger RNA, right? So messenger RNA, mRNA itself. So what is this messenger RNA? It forms totally 5% of total RNA in the organisms. So out of 100% of RNA, 5% is mRNA. 5% is mRNA. It forms 5% of total RNA, and it is single stranded. It is single stranded and is synthesized on one of the strands of DNA. It is synthesized on one of the strands of DNA. Say for example, if this is the DNA structure, okay, and this DNA means it separates at a certain region. Both of themselves separate at a certain region like this. Let it be like this only. See here the same DNA. This is 3 dash, okay, and this is 5 dash. This is 3 dash. This is 5 dash. This is the DNA, and this DNA. Sometimes what happens over here is the strands that separate like this. See here, that separate like this. So when it is this strand that separates, so on one of the strands, the bases that you come across over here, on this A, G, C, T, right? A, G, C, T, like this, all the bases will be there. Single strand of DNA it is. On this single strand of DNA, there is synthesis of mRNA that takes place, which is being complementary to this DNA strand. Okay, so this strand is being complementary to this strand, and this strand is 5 dash to 3 dash, and this one is nothing but mRNA. What it is? mRNA, which is being complementary to this DNA, but only thing is thymine is replaced by uracil. Thymine is replaced by uracil over here. So this is how the synthesis of mRNA that takes place on one of the strands of DNA, remember, right? So this is how the mRNA is synthesized on one of the strands of DNA. And this mRNA consists of a message for the synthesis of one of the proteins. The required protein in a cell inside the nucleus, what is present? DNA is present. And this DNA, that both the strands that separate at one point, it is called as origin of replication. So at this region on one of the strands, the synthesis of mRNA that takes place on one of the strands of DNA. So that's why mRNA as well as DNA are being complemented to each other, so that it can copy the message from the DNA. So on the DNA there is a message, coded message is there, and that is copied by mRNA. According to that message, what it shows is, it consists of a message for the synthesis of protein. According to the message what is present in messenger RNA, so itself is synthesized one protein. Okay, so this is how mRNA that is synthesized on one of the strands of DNA, and it comes from the nucleus to the cytoplasm. It comes to nucleus to cytoplasm and meets the smaller subunit of what? Ribosome, that is the TS in case of prokaryotes, and 40S in case of what? Eukaryotes. So this mRNA that comes and it binds to this what? It binds to the smaller subunit, and later with the help of the Mg++ ion, so the larger subunit 60S is attached over here, and this is, since it is called as protein factory, so there the synthesis of protein molecule that take place according to the message present in the mRNA, right, inside the cytoplasm. There the synthesis of protein that take place over here. So now we are very clear that this is one type of RNA which does not act as the genetical material. Hence it is non-genetical RNA, but it helps in what? Synthesis of protein. Right? So this is the fact to be remembered over here. So this is one thing regarding messenger RNA. And the next RNA is trRNA. Next one is trRNA. It is commonly known as transfer RNA. It is the second type of non-genetical RNA which is not acting as the genetical material of any organism, hence it is called as non-genetical RNA. It is also one type of RNA which is synthesized on one of the strands of DNA. It is also synthesized on one of the strands of DNA, and it forms totally 10 to 15% of total RNA. It forms 10 to 15% of total RNA over here, and this RNA, it is called as transfer RNA. So why? Because it helps in the transfer of amino acids. It helps in transfer of amino acids. So from cytoplasm to region of protein synthesis, right? From the region of cytoplasm to the region of protein synthesis, that is nothing but up to the ribosomes. Ribosomes are called as protein factories. To these factories, the raw materials, that is amino acids, are carried by this transfer RNA, tRNA. So, and this tRNA, is also single-stranded. Is also single-stranded RNA, but it folds on itself to form double-stranded here and there. Here and there it becomes double-stranded, right? So because it folds on itself, so here and there in its structure, hence it becomes double-stranded sometimes, right? So, and this tRNA, as I said, it is also synthesized on one of the strands of DNA. So, and this tRNA has clover leaf model. It looks to be as clover leaf or a leaf of the clove plant, Hence it is said to be as clover leaf model of tRNA. So model of tRNA, it is said to be as. So as you are seeing there, this is the structure of tRNA. Looks to be as clover leaf model. Okay, here it has become double stranded, here it is single stranded. Single stranded over here. Here also it is single stranded. Here also it is single stranded. Okay, so in all these three regions, there are single strands that you come across, and these have some arms. See here, so here the amino acid that binds over here, that's why it is the site where the amino acid are binded over here. Hence it is said to be as amino acid binding site, it is called as. Amino acid binding site, it is called as. So there are three loops that we come across over here. So DH loop, T-Ci-C loop, as well as so anticodon loop. Like this, there are three loops that we come across. Along with that one more loop is there, that is variable loop is there. Variable loop is there, and this is called as a T-Ci-C loop. It is called as, and this is called as a DHU loop. So like this, there we come across all these structures in a tRNA. Okay, so this is about the tRNA and which help in carrying the amino acids. See here, there are 20 types of amino acids are so participating in synthesis of particular proteins over here. That's why for carrying one particular amino acid, there must be one particular tRNA. That's why totally there are 20 types of tRNAs are there. So for carrying 20 varieties of amino acids. So what it shows is, so here the amino acid is specific for a specific tRNA. So both are specific to each other. That's why so these tRNAs are 20 types. Right? So I shall write that point also. There are totally 20 types of amino acids which are used for synthesis of different proteins over here. Different proteins over here, and that is why so 20 types of tRNAs are also there for carrying one specific 20 specific amino acids here. So what it shows is there is a specific tRNA helps in carrying specific amino acid over here, right? So this is the explanation regarding the tRNA, and the last RNA itself is ribosomal RNA, that is rRNA. Yes, rRNA. It is called as ribosomal RNA, and this ribosomal RNA, which means it forms a component of ribosomes, a major component of ribosomes. Why? Because the ribosomes are made up of one is protein, and they consisting of rRNA. So that's why rRNA forms the major component of ribosomes, right? So next one, this rRNA forms. So see here, one more point to be noted regarding this tRNA. It is the smallest RNA. Smallest RNA, right? That point to be noted. And this is maximum 70 to 80% of total RNA forms. 70 to 80% of total RNA it forms. I mean rRNA that forms over here. And this rRNA which is being complementary to the mRNA, right? So see here, rRNA is being complementary to mRNA, and it is also synthesized on one of the strands of DNA itself. It is synthesized on one of the strands of DNA itself. So that is nothing but rRNA, right? So this is how three RNAs that play very important role in protein synthesis and all other activities inside the cell, right? So these are non-genetical RNA since they do not act as the genetical material of any of the organisms, hence they are called as non-genetical RNAs. So this is about the nucleic acids what we have gone through. So first one is DNA and second one is RNA. So RNA types are also studied over here. So by this we finished one more biomolecules, that is nucleic acids, and the next one is proteins. So what are these proteins? How these are synthesized? So what are the types of structural proteins? All these are the things, and their biological significance are also studied over here. Now we shall start with the next biomolecule, that is proteins. Now proteins. What these proteins are? So the question arises, what are they made up of chemically? What these proteins are made up of? Who discovered these proteins for the first time? Right? So these are all things that we study. And what are these proteins made up of? So whether these are made up of the chemical components. So what chemical components or what structures these proteins are made up of? Simply you know it better. It's a polypeptide chain. It is also called as, what it suggests, it is made up of many number of amino acids, so which make a long chain of amino acids, so together form the proteins, right? Simple. This much of basic knowledge what is there with us now, right? One of the scientists called Mulder is the name of the scientist. Gerardus Mulder is the name of the scientist, the one who discovered these proteins itself for the first time. So Mulder is the name of the scientist, the one who discovered. So Berzelius is the name of the scientist, the one who coined the term proteins, right? Berzelius is the name of the scientist, Mulder is the name of the scientist, the one who discovered these proteins for the first time. And these proteins are, totally they make 2/3 of the entire cell. 2/3 of the entire cell is made up of these proteins only. Okay, and so these proteins are chemically made up of proteins. So they are made up of what? They are made up of C, H, O as well as N. So these are the elements mainly that makes these proteins. Along with this CHON, so ultimately sulfur is also another element present over here in the proteins. Sulfur is also present over here. Then what these proteins are? What is the definition, actual definition of these proteins? We can go through. So these are nothing but heteropolymers. These are called as, why they are called as heteropolymers I will explain, right? So these are the heteropolymers of repeating units called as what? Amino acids, right? So means the proteins are simply, they are made up of what? Amino acids. So that's why we shall define them as, so these are heteropolymers of repeating units of amino acids called as proteins. Simply we can imagine this one. So this is one amino acid, this is another amino acid, this is another amino acid, this is another amino acid. So like this, a chain of amino acid is called as polypeptide. Or these are known as, so polypeptide. Poly means many, peptides itself is the bond. This is the peptide bond. This is the peptide bond which is present between two amino acids, right? Such kind of amino acids which are bonded with the help of the peptide bond so as to form polypeptide chain, and this polypeptide chain itself is nothing but it is a protein molecule. It's a protein molecule. Chain of amino acids is called as polypeptide, and it is also known as nothing but, so it is proteins. So now we came to know what these proteins are made up of. They are made up of simply the amino acids. How many types of amino acids these proteins are made up of? The question arises. See here, so naturally there are more than 300 number of amino acids are there. They are existing. Out of 300, 20 types of amino acids are used in the synthesis of different varieties of proteins itself. Thousands of varieties of proteins are there, and they are made up of, so sequence of different types of, so these amino acids. When the sequence changes, the protein changes, right? So this way the proteins are made up of a sequence of amino acid or the chain of amino acid, right? So there are, simply you write here, there are 300 more varieties of amino acids are there, out of which 20 amino acids are used in the synthesis of what? The proteins. So they are used in the synthesis of proteins. Already I have told you there are 20 varieties of amino acids that are carried by the tRNA, right? So that is why there are 20 varieties of tRNAs are there for carrying 20 varieties of amino acids. It shows that one amino acid is specific for one tRNA, right? So that I have mentioned it. Okay. Such kind of 20 amino acids are used in the synthesis of these proteins. Okay. Yes. These proteins are mainly synthesized in protein factories. Protein factories. Those proteins factories are ribosomes. Factories are ribosomes. Ribosomes help in synthesis of these amino acid chain, that is polypeptide chain, into proteins itself. There are different types of properties. Properties are there for proteins. Those properties are very important to study. If we know the properties of these proteins, then we can understand how these proteins are. So how these proteins are made, right? All these are the things basically we can understand. So what are the properties of these proteins? Let us see now. So the first property of these proteins is, so there are thousands of proteins that make the body of the organisms. Right? There are thousands of protein varieties that makes the body of organisms. Right? So, and they are showing the specificity. So these proteins, what are forming the body of these organisms, they are specific. They are specific one, so that they can show specificity, right? They can show specificity, right? So this is one characteristic feature of these proteins. So these are made up of one or two polypeptides. What are these proteins made up of? Proteins are made up of one or two peptides, right, or polypeptides, yeah, made up of. So right, so such kind of chain of amino acids together form what? These proteins. These peptides are formed as a result of polypeptides, which are made up of chain of amino acids bounded with peptide bond. They are bounded with peptide bond. Already I have shown you. So this is one amino acid and this is another amino acid. So both are bonded with the help of peptide bond. So this is also one more characteristic feature of these proteins. Third one, these proteins have high molecular weight. High molecular weight. So these proteins have high molecular weight is also another characteristic feature of these proteins. So these proteins are not easily passed through the membrane. This is very important. So important feature, these proteins which are having high molecular weight and larger in size. So these proteins are not easily passed through the membrane. That is nothing but plasma membrane. Through the plasma membrane, they are not easily passed through. So even though they have to pass through the membrane, then there occurs some of the active transportation like endocytosis as well as exocytosis. Through endocytosis as well as exocytosis, they have to pass through proteins, even though wants to pass through the membrane, then so they have to pass through by endocytosis or exocytosis. So any one of the method they follow to pass through the plasma membrane. So this is also one more property of these. So what? Proteins. And next property of these proteins is these proteins can easily dissolve into the water, right? So they dissolve into the water. Sometimes they make the colloidal systems. These are sometimes soluble in water and sometimes they make colloidal system. This is also one more property of these proteins. Next property of these proteins is, so these are amphoteric in nature. Proteins are amphoteric, or the amino acids are amphoteric in nature. So that's why simply indirectly we can say these proteins are amphoteric in nature. So here amino acids are amphoteric in nature. Means what? These may have different reactive groups. They have. They may have COOH group, right? Sometimes these may have amino group. Such kind of groups, reactive groups, these amino acids have. Hence, they are said to be as amphoteric in nature, right? This is also one more property of these proteins. And the next one is, so these are denatured. When proteins are denatured, by any one of the methods, when they are denatured, means so change their structure, denatured, so temporarily or permanently. So when proteins are denatured temporarily or permanently, so when they lose their three-dimensional conformation, they are denatured. So when proteins are denatured, so temporarily or permanently, so when they are denatured, when they lose their three-dimensional conformation. So these are some of the characteristic features of these proteins. Okay, so by this we finished one part, the general characters or the properties of these proteins now. And after that one, so let us Let us see here in case of proteins. So these are the biomolecules which are made up of the monomers called as amino acids. See here, these are amino acids, these are proteins. Amino acids together form proteins. These are said to be as macromolecules, and these are said to be as macromolecules here. So first of all, we will have to study these biomicromolecules now, and afterwards we will have to study biomacromolecules. That is nothing but proteins. So that is why amino acids. What these amino acids are, let us study in the next video. Okay, till now what we saw is, so what are the types of RNAs? So one type of nucleic acid, that is RNA, we finished in this video, and after that one we started with so proteins. Proteins are also the biomolecules which are made up of biomicromolecules, that is amino acids. And what are the major properties of these proteins, so that we saw in this video. In the next video we will start with the amino acids. Thank you.